Hi, I was getting my last tweet out there on Sasha's talk. Glad to see all the Twitter activity and everything going on. So we're really getting out to the world with this. Really fun to see. I'm Julie Graylow. I'm the Director of Breast Medical Oncology at the SCCA, the EW, and Fred Hutch. For the next two pieces of this, we're going to have panels and break this up a little bit. So the first panel is going to be on how laboratory research can help patients. And I'd like to invite our panelists up. We have Kevin Chung, Corey Painter, and VK Gotti. And you've already met VK. I'm going to I think what we'll do is, I have just a couple introductory slides, and then Corey has a really nice slide set on the Metastatic Breast Cancer Project, which is totally amazing. And then we'll ask Kevin and VK to tell us a little bit about their lab work and how it could relate and get into patients and why that's important. So how do I now get to mine? Here we go. So here is our panel with VK pointing to his SCCA hat. Just a couple introductory slides from me. You may or may not know about our U.S. Precision Medicine Initiative, which is a national initiative kind of launched in his State of the Union address in 2015. President Obama launched a general precision medicine initiative, and as part of that, cancer and oncology was felt to be a key driver of where we should be focusing our precision medicine efforts. So the bulk of the money that went along with this went to oncology and the National Cancer Institute. So since we're talking about laboratory medicine, I'll show you the four key components to our National Cancer Institute's Precision Oncology Initiative. The first is that they want to expand precision medicine clinical trials, assigning therapies to patients based on their genetic or their tumor's genetic alterations in this case. They want to focus efforts on overcoming drug resistance. We all know in metastatic breast cancer that many times the tumor responds to a therapy, but then it figures out how to grow through it. So we need to research more on how tumors become resistant to therapies. And then number three is all about laboratory research and how it's critical. And in particular, they want to focus on response to treatment, but all kinds of things that we really need to study in the laboratory in order to figure out what might work and get hypotheses to test in patients. And then the last is the development of a national cancer knowledge system, a database for integrating genomic alterations from tumors with responses and patient outcomes. So this is what we're talking about here at this panel, going from bench to bedside.
So I believe in this concept of translational medicine, which is a physician scientist in the middle, looking at things not only going from bench to bedside, which is what we say for ideas that are cooked up in a laboratory and somehow make it to a patient, but that there's an arrow missing. It's actually also bedside to bench. We learn things from our patients. We generate hypotheses treating our patients, and we go back to our lab. And so the role of physician scientists is that's what it is. I mean, Kevin and I are both physician scientists. We actually have our labs right next to each other, and we get to see each other every day. But we also get to take care of patients, and it's truly an honor to be in that kind of ground floor. And I'll just give you one other sort of plug for why people like us need to be involved and also engage our advocates. I had a great, really brilliant biomedical engineering student join my lab 7 years ago. And he was working on something called high-intensity focused ultrasound. So he basically took ultrasound waves and liquefied tumors to generate an immune response that would then kill the tumor. And we started doing this work, and after a while, we kind of all threw up our hands and said, this just isn't going to be a way forward. And he said, why? All the tumors are dead. And we all looked at him and said, yeah, but so are the mice. So we have to have people like us, as well as advocates in the middle, looking at this bedside to bench directions to figure out what the right hypotheses are to move forward. And that's really what the role of physicians like us is.
Yeah. Great. I'm Kevin Chung. I'm also a physician scientist and assistant member. I've actually been here for a little over a year. And Fred Hutch is a fantastic place. There's a lot of great physician scientists and a lot of energy here, which is fantastic. I want to echo that, in terms of thinking about, on the lab side, there's so many questions you can ask, so many directions you can pursue, but as a physician scientist, it's very clarifying. You think, where will this go? Where can this lead to making a difference for patients? And so that's something I wake up every morning.
Hi, everybody. I am a scientist, but I'm a patient, so I guess I would be a patient scientist. So I'm a physician scientist. And I'd like to start by thanking so many of you who I know, because you're part of the MBC project already. And I see your faces. I'm Facebook friends with most of you. I tweet with most of you or email, and it's just amazing. And what I'm going to talk about is the power and the absolute need to partner directly with patients in order to truly accelerate the pace at which we can make discoveries for the genomics research that we're conducting at the Broad Institute of MIT and Harvard, where I work. I'm going to fly through these slides as fast as I can, but I'm happy to email any of you a copy of this slide deck or talk with you during the break, or if you open up your packets, there's actually a brochure for the MBC project where you can find our information. So don't worry if if I fly through too fast. I promise I'll get to the information. So with that being said, where does this slide begin? Okay, so this study that we're conducting at the Broad is led by Dr. Nick Wagley, who's just a phenomenal oncologist and genomicist. And he conceived of this idea in order to really address these questions, these laboratory translational questions. What are all of the molecular changes that lead to metastatic breast cancer? Why do some patients have extraordinary responses to some therapies and others don't? Why do some tumors never respond or seem to have resistance to all therapies from the very start? What leads to developing metastatic breast cancer at a very young age? What genes are involved in metastatic breast cancer in underrepresented populations of people? The vast majority of what we've studied to date as scientists are Caucasian people, and that doesn't cover the story for the people that actually develop this disease. How can we improve the use of genetic information to treat metastatic breast cancer? And after we get the answers to all of these things, how can we develop better treatments for people that have metastatic breast cancer? Part of the problem with why we have not been able to address a lot of these questions to date rests on where tissue to study even comes from. So the vast majority of people who get metastatic breast cancer, or any cancer for that matter, are treated in a community setting. Only 5% of people are enrolled in clinical trials, which is where we get most of our tissue. So if you're in Idaho and you have a resection or a biopsy, that tissue is used to confirm your diagnosis, and then it goes and sits on a shelf somewhere. But what happens if we were able to leverage that tissue and get access to the vast majority of tumors that are just sitting on shelves places? Everybody's tumor probably holds some unique characteristic to their biology. And if we could really look deeply at a large swath of what happens on that individual basis across many, many people with metastatic breast cancer, and then look at what their clinical course was, we could probably answer a lot of those questions that I posed in my first slide. And we live in an era now where it's possible to do all of this, right? We all have, how many here, just raise your hands, do you have a smartphone? Let me see. Oh, look at that, right? So we all basically have smartphones, we have internet access. This wasn't the case 10 years ago, but now it is. And we should be taking advantage of all of these things in the way that we're all interconnected. I am, as I said, a cancer patient myself. The very first thing I did was find other people like me. And I think that holds true probably for the vast majority of other people with different types of cancers. We find people like ourselves and we take comfort in that. But we can also find people like ourselves and drive research as a result of that interconnectedness. And that's one of the things that we're trying to really leverage with the MBC project. So what we're doing And this is kind of our approach, was we got a protocol together, and you don't have to read this, I'm just gonna say it really quickly. We have a consent that's online, so you can be in the comfort of your own couch in Idaho, it doesn't matter. And what we do is we ask for permission to get medical records, some of your stored tumor sample that's sitting on the shelf, not being used for anything, a little bit of saliva, and there's a lovely new video of Dr. Wadley actually spitting. If you would like to see how this happens, it's really great. I mean, it's making spitting great again, so you should go and check it out, it's on our website. And then we do deep molecular characterization. So we look at the DNA, we look at the RNA, and again, during the break, happy to talk about the details of this with any of you. And then we do that next layer of taking the medical records and looking at the responses and clinically annotating that data. And what we're trying to do with this is make it publicly available to other researchers. We don't want to silo off this data. That's a huge problem, which I think anybody that's in the medical profession and beyond appreciates. That, the typical cycle of I get my data, I publish my paper, you guys see the tip of the iceberg, and then I hold the rest. We have to do away with that if we're going to make big, sweeping changes. And that's really, at the nucleus of what we're trying to do here. We want to make those discoveries and publish, but we want everybody to look at it and make their own discoveries. So, those of you who are part of the project and the solution, who are following in social media or getting our email updates, see that we routinely put our aggregate data out there for everybody. So everybody knows where we are with the study, what we're learning, and right now we're starting to do the sequencing. We are going to rely on a lot of the patients that are part of the project to help us interpret that and message that. So here's a look at the website. I'm not going to go too into it, but we developed everything in lockstep with the patients in the community, including one sitting right here in the front row. And we've had over 2,800 women and men sign up for the study from all 50 states. This is just a quick look, and again, I'm not going to go over this in too much detail, but of the way this is truly driven by patients. Patients really, because they helped us build it, were stakeholders from the very get-go. Right? And they were the ones to tell us, this is what you need to do and this is how you need to do it if you want to be successful. And we listened to them, and then they took it to town. And there were all these different instances where you see these stochastic rises in registration as a direct result of patients who were involved. We then started sending out consent forms and saliva kits, and they've been pouring back in. And yes, that's a really bad time. You can't hold it. And what we have ended up with at this point is really rich, detailed patient-reported data that we're verifying through actual medical records. But what we, I think, maybe didn't even fully appreciate when we started the study was when we ask our, couple questions at the get-go, patients are able to self-report very interesting information that would take years for us to go through people's medical records and find for ourselves. And we didn't, I don't think we really fully appreciated that. That's the truth. And as a result of that, if you look through what patients have told us about themselves, they have allowed us to build out individual cohorts of people that have had extraordinary responses to therapy, which is an exceedingly challenging thing to find in somebody's medical record. Each of these groups now is readily identifiable as a result of the questions that patients have provided answers for. So, for example, we ask, have you been on a therapy for 2 years or longer? And have you had an extraordinary response? And people say yes or no, and if they say yes, 99% of those who say yes tell us which drugs they were on. So bam, right there, we can just go and find the people who have had extraordinary responses to capecitabine or platinums or everolimus. And right there, then we just go and we can verify through their medical records, and sure enough, they know what they're talking about. And so we can then prioritize those patients to sequence, and instead of doing thorough abstractions from 2,800 records, we can start right off the bank from people that are going to have insight for a lot of other people with this disease. Patients are driving this disease, but they're not numbers. They're so dear to our hearts, and it's touching to every single one of us who is involved in this project. When they tweet pictures of their saliva kits after they get them, and all of us just get so jazzed. The IT people at the Broad Institute are like, look at this, They're really excited. They feel so connected to each one of you. The scientists who receive the samples at the genomics platform, who never get to see patients or see the end result of the great work that they do, take so much pride in their job now that if people decorate their kits, they take the tops off and they have like the wall of fame of notes that came back, of like people that had their kids decorate them. So impactful and so meaningful. It's really lovely. And as a result of this amazing patient response, the Broad and others have taken notice. I mean, the NBC project has gotten national attention. And people are like, wait, what? Patients? Look at that. And so everybody wants to see more of it now. And so the Broad has taken this initiative and allowed us to run in other directions with it as well. And so we're getting ready to launch the next project, which is modeled after the NBC project, called the Angiosarcoma Project. More about that later. And I just really want to say thank you to all of our advocacy partners who played a huge role, including one of our newest members, Susan Coleman. So we would like to thank them. Patients of the driving force here. And with that, I just want to say thank you to everybody.
It's a great project, and I hope if you haven't participated, you now will be excited to participate in this project. So Corey, I'd like to pass this over to Kevin briefly and have you tell us what you're doing in your lab, because you're doing some pretty cool stuff. And Kevin is, both a physician and a scientist. He is a medical oncologist, been here about 2 years.
What's a year and a half?
Year and a half.
Yeah.
So, but he is a medical oncologist, and so he has both the clinical and the laboratory perspective. Tell what are you doing in your lab?
Thank you. Yeah. So my lab studies how cell clusters metastasize, and what I mean by that is that, so you may have heard maybe from doctors or been reading about it, metastasis is often thought about as individual cancer cells that have broken away from the breast and then seeded to some organs. Well, what we've discovered is there's, in fact, much more to this story. And in fact, cancer cells really like to travel as a team, as a cluster of cells. Now this matters because we found that these clusters can seed metastases over 100 times more efficiently than single tumor cells. And so the vision of my lab is that by studying the behaviors of these clusters, studying their weaknesses, we can unveil new pathways that could lead to better therapies for metastatic breast cancer patients. Now, 100% of what we do in my lab is focused on metastatic breast cancer. And so we think deeply about how understanding this biology could lead to new ways, new types of therapies, or better approaches to therapies that we have. And so, as an example of that, we've been studying clusters in the lab, and what we found is that these clusters are incredibly resistant to cell death, a form of cell death called apoptosis. And what we've discovered now is that the cells in these clusters are talking to each other. They're communicating by these signaling molecules which we don't understand fully yet. And we think that that might be a new key to different kinds of therapies that don't exist today. Now, at the same time, we're also looking at therapies that are being given to metastatic breast cancer patients. And what we're finding is that in some cases, these therapies may not work for some of the cancer cells that are within these clusters. There's different types of cells in this team, some more or less resistant to treatment. And so what we hope, these are early days, okay, but what we hope is that this will lead to better therapies and ultimately ways to prevent metastatic breast cancer from ever even happening.
Yes. Which, of course, we prefer never to have to deal with metastatic breast cancer in the first place, but we've got all these people dealing with it, so we have to work at both ends of it, too. So VK's doing some really interesting laboratory research as well. Why don't you tell us about what you're doing in your lab?
Sure. Before I do that, Robin just reminded me, if you guys have questions, there's little note cards in your folders. You can write them down and pass them to the end, and people will be picking them up on the end so we can answer those questions. This is a Q&A section. So my laboratory is a little bit older, and there are a couple different focuses in the laboratory, but I think I'll focus on one that's probably most relevant to metastatic breast cancer, which is, for years now, we've been doing genomics and trying to understand how all these mutations are put together inside a cancer cell that might lead to the cancer cell behaving the way it does. But to some respects, that's a very static view. That's what that cancer looked like at that moment. And we don't understand necessarily how all these things fit together. And really to get a better understanding of this, the next level, the next sort of thing that needs to happen is a functional understanding. How are these genes put together and sequenced in a way that these outcomes happen within the cancer cells? And so what we've been developing over the years is a way to culture cells from patients, grow them in the laboratory, retain the features of those cancers as they would be in the patient, except in the laboratory, and then come back and knock out one of these genes at a time to see how the cancer cell is really constructed together. So the way I think about it is the following. How many of you actually got here today using a paper map? Right. Well, that's what we have for cancer. We have one person. I won't point out where she is. No, so most of us don't do this anymore because that doesn't contextually tell us a lot about traffic, the weather, and so forth. So how many got here using a smartphone with a Google map? Yeah, and that's how most of us work nowadays. Yet we don't have that for cancer. What we have are basically two-dimensional maps of where these mutations are and so forth. And if we can build these functional maps, understand where the blockages are and where the open pathways are, then we can block these things with new drugs in ways that we weren't able to do before. And this is really kind of what my laboratory has been focusing on specifically in metastatic breast cancer.
Good. Great. So as you can see, just a small example of some of the research going on in the lab. I hope you can see that it directly applies down the line to what we might have in order to treat metastatic breast cancer or prevent it in the first place. So Linda, I see you have a few cards, and if you have questions, pass them on down. So here's a metastatic breast cancer question for Corey. Are you asking the question of complementary and alternative medicine use, and if so, how are you quantifying?
That's a great question that we get often. So we have in our surveys sections for people to write in anything that they think is important for us to know about. We don't specifically ask about all the different types of supplements that people could take or all the different types of lifestyle alternatives. However, people are self-providing that information. We have to focus our studies in the areas that we have specific scientific expertise right now at the beginning, but it is an evolving project, and should we bring along an epidemiologist, all of that data is there, and we'll be able to address these questions. So we don't have a good way to quantify a lot of these things. I'm asked, how are you going to figure out if stress caused my cancer? And to be honest, as a scientist, I don't have a good metric for being able to come up with something that would allow me to quantify something like that. And so right now we are focused on the genomics, but hopefully at some point we will get to a lot of these other things that we all know are very important to consider.
So as a follow-up, a couple of related questions. One is about how many different groups are looking for participation, like Dr. Susan Love's network and the Young Survival Coalition. So how is data shared across these groups, et cetera? And then another patient is asking about that she doesn't have metastatic breast cancer, but had breast cancer 10 years ago. Can she join this study, or are there some of these other groups who are collecting information and samples from those patients?
Those are all fantastic questions. So we are partnered with a lot of the different organizations like Susan Love, and are promoting the studies that they're doing, and they're promoting the studies that we're doing. In terms of data sharing, one of the things that we're trying to do is set the standard for doing it in the first place. And so we are actively looking for a variety of different places and ways to house our data, including generating our own database so that this can happen. I cannot speak to any of the studies that our partners are doing in terms of how they're going to release their data or what they're going to do with it. But what we'll do is we will come up with ways to share in a variety of different platforms, like the Genomics Commons, like with our own home database that will have access for all biomedical researchers. In other unrelated news to the NBC project, we have the Broad in general is looking at collaborations with the Hutch that, in terms of having the Hutch host data, and then we have our capabilities that will feed into it so people can use the analytical platforms. So there are a variety of different ways that at the Broad in general, we're looking at this question of data sharing, and we're hoping that we can kind of lead by example to show that impact of how great it is to share the data, and then everybody will hopefully join on that bandwagon.
Great. Great, thanks for that. A couple questions on the lobular subtype of cancer. So Kevin, one of the questioners points out that lobular cancer doesn't tend to cluster so much. So how might your research impact invasive lobular versus a ductile subtype?
That's a really an interesting question. Yeah, so I think there's a lot more to be learned about lobular breast cancer. If you think about from medical school, when all medical students learn about lobular breast cancer, it's this single file chains of cells that are kind of maligned. So there probably is some cell adhesion, cell contact between these cells that might be important. That's it. I think that we're interested because we've also seen in our research certainly that sometimes we can look at ways to allow single cells to survive better. And so we're interested in ways of developing therapies towards that form of invasion as well. So it may be relevant.
Great. VK, the next question is asking about how technology is assisting your research.
Greatly, I would say. And this really goes to Corey as well. The kind of data we get is ridiculous. It is way more than you can keep in any one brain. And so where it's really made a big impact is as we have better and stronger computational abilities, we are able to crunch through this data more efficiently and get our answers quickly. They're not specifically related to my research, but there are people that design proteins and things like this where rather than just having the lab where they're located crunching out all of these things, they're actually sending parcels of programs to people's computers at their homes, and when they're asleep, the computers are actually churning out data and feeding that data back to them to help them design proteins and things like this. So there's a lot of technological innovations going on from the patient perspective, from the companies that make these computers and programs, supercomputers, et cetera. We actually have to use all of those kinds of things, but really it's become incredibly data-dense, data-driven. And there's a lot of pitfalls as well with that. One of our great colleagues, George Sludge, who's the head of the Cancer Center down in Stanford, he has this great thing. He says, if you torture the data long enough, it'll tell you something. You have to keep that in mind because this data can be very complex and you can misinterpret it as well. So I think those are the ways that technology, of taking this question correctly, is really impacted my laboratory.
Great. Some of these questions, I'm realizing now for next year, Linda and Beth, we need to have a Q&A after the presentations we had first because some of them are more directed to those presentations. So I'm going to save those and see if we can find a way later in the program to get to some of those.
And I'll point out some of the speakers actually left, but there's a flight that some of them had to catch.
Yes.
But we still have Sasha and Julie.
Yeah. So I'll try to get, we have about 30 seconds left in the session too. So I'm 5 minutes. 5 more minutes? Okay. My battery on my watch stopped yesterday, which I think is nature's way of saying I just need to slow down and not pay attention to time so much, but when you're moderating a panel, you do have to pay attention to time. So we have a few more minutes. So I'm going to try to pick questions most related to the people who are sitting up here right now. And one question I think is very relevant to lots of people in the room. I'll ask VK and Kevin, in addition to participating in the Metastatic Breast Cancer Project, how can metastatic patients initiate donation of biologic materials for basic science research at the Hutch?
Okay, yeah, sure. And so I think that as VK had brought up the idea of, working with actually the patient's own tissue and then studying that in the lab is very valuable. And I think an important analogy is if you watch football, you're watching the game. If you're from outer space and you don't even know how the game works, if we just took those players and look at their molecules or look at their genes, would we understand how it works. And so by studying in the lab and, okay, all right, we can observe it, use microscopy, we can use functional genomics and other approaches, we can sort of get at how does metastatic breast cancer work and drill down to better effective therapies. Now, in my own lab, what we are very interested in doing, we've made developments of organoid models. These are, you can think of these as mini tumors that we can culture from patients. And what we do is we study these organoids in 3D systems that model what's going on in a patient. And through these studies, we've discovered cells that are involved in invasion and metastasis, and we're using this system in which that attack from many different angles the vulnerabilities of these clusters. And so, for us, we're very interested in obtaining patient samples and making these organoid models that we can study and then really build on. I think.
And I'll just say, when you go in to see your clinician, ask, is there a research study for me that I can participate, where I can donate my tissue? Unfortunately, we're not, for ethical reasons, we're not in a position where we can just randomly grab tissues off the shelf. We really do need to have your permission, and we have to use those tissues in a way that respects your privacy and all these other things. Corey's broken down a lot of these barriers to make this feasible, but there are individual research studies at all these different hospitals. Even in Seattle, most of the hospitals do research here, and you just have to ask and say, is there something I can have this tissue be put to words? And we will obviously want to do that. And most of us are tuned into this, and we will always ask as well.
So I will say, we have a good mechanism for collecting early stage breast cancers within our system, because when we see an abnormal mammogram that looks highly likely to be cancer, the patient is consented before her first biopsy for some extra samples that can go into a bank. So we have a good way of identifying those patients and getting that. We have absolutely no mechanism in place right now for getting a collection of metastatic samples. In many ways, it's related to funding, frankly, because we would need the study coordinators and the collection and the processing and all. This is important. It's frustrated me for a long time that we don't have a metastatic bank of tumors, and tumors that are serially collected, because we know that over time with exposure to different treatments, the tumors change their genomic profiling. And I think this is something that we just really need to kick up the interest in and say, this is actually kind of embarrassing to me that we don't have the collection of the metastatic tumors, and the patient voice can really help with that. So if you stir it up and say development, help us fundraise, or, this isn't the kind of thing that's easy to write a grant for, frankly. It's not, because the idea is you're getting samples for everybody to use, and you don't even know what you want to do with them yet. And the grants require that you actually have a hypothesis for the most part, and you know what you want to do with them. So the collection, the money that we pay for the collection of a metastatic bank here would actually not be through typical grant mechanisms. So I have less than 1 minute now, and let's just end again with some questions for you, Corey. One of which is, I sent in my sample, or I sent in my request and I haven't got my sample yet. What's the turnaround time? Another person says, looks like you sent out 200 kits or so, 2,000 kits, but you only got 900 back. What's the delay, or how can we get people to send their kits back sooner? I think if we can address that in the last few minutes, that would be helpful.
Yeah, sure. So thank you for asking these questions. And I think that people, one of the things that I want to stress is that we are a research study. So we're not like a company with a lot of people that get a consent and then that day ship something out by FedEx or anything like that. We actually needed to know quite a bit about who is going to respond, if people were going to respond, what the response rate was going to be. And so if you remember one of the slides I showed, I had kind of this rise in people that were signing up. And then we waited several months before we even shipped out our first consent form, because we weren't sure if this was going to work, to be honest. We needed, the whole notion of patient engagement was in and of itself an experiment. So we needed to make sure it was going to work. And it did, and it worked amazingly, and it's still working phenomenally. And so we had a couple month delay. We started sending out consent forms.
with those consent forms, we sent out, so to date, 2,800, and I think about 1,700 people have filled out the actual consent. Then after consent, that's when we started triggering the saliva kits. The saliva kits we started sending several months ago. We've only sent out to date about 1,300 saliva kits, and we've received back 900. And so that's actually beyond our wildest expectations in terms of saliva kits. Saliva kits will be sent to everybody who provides consent. So we're just, just a little bit of a lag as we continue to ramp up. But right now, everybody who signs up gets a consent form. A lot of people, when they get the consent form, the whole point of consent is for people to really read through and make sure they are comfortable with all aspects of the study. And a lot of people, when they read through the consent, they're like, oh wait, you need my tissue? I don't know what that means. I might not be comfortable with that. And some of them will call us and I'll say, please do not sign consent if you're not comfortable. The whole point is for you to really understand what it is, be comfortable with it, and then sign consent. And so we have, well over half the people who have signed up and provided self-reported data provide us with that consent. And for that, it's again higher than I think we had anticipated. The return of the saliva kits is amazing. I am sorry that there is a little bit of a delay if you sign up in terms of getting your saliva kit, but so far so good in terms of everybody's collective efforts with this great group effort that we're trying to put together.
Great. Let's give a big round of applause.